Hello, microbiology students. I hope you guys had a good weekend. We're now in week 2 of Bio 113, and we are still talking about the structure of bacterial cells. And today we are going to talk about the cell envelope, the outer layers of the cell. So let's go back. So we talked, just a quick refresher, on Thursday. We talked about the nucleoid. We talked about other things in the cytoplasm, including, structures like gas vesicles and magnetosomes and inclusion bodies. And we talked about cytoskeletal elements that are involved in cell shape and in cell division. So today we are going to move to the layers of the cell moving outward. We'll start with the cytoplasmic membrane, and then we'll go to the cell wall, and then to the outer membrane and surface of the cell. Okay, so let's talk briefly about the cytoplasmic membrane. So bacterial cytoplasmic membranes are made up of largely the same kinds of phospholipids as eukaryotic cell membranes are, not necessarily the exact same species. So our classic phospholipid has a hydrophilic polar head group here. You see the phosphate group here. The constituents out here can be variable. And then we have the hydrophobic fatty acid tails. These are the hydrocarbon chains. They can be saturated, as you see here, or unsaturated, as you see here. The length of these can vary, so not every phospholipid molecule is the same, but this is the general structure. And of course, in a biological membrane, we create a hydrophobic interior as the fatty acid tails here face each other for each layer of the bilayer, and then on the outside facing the aqueous environment outside of the cell, in this case, are the polar head groups, or inside of the cell facing the cytoplasm are the polar head groups. And then very important to recognize that there are many proteins embedded in this phospholipid bilayer, some of which go all the way across, some of which may be only sort of embedded in the inner part, or even potentially in the outer leaflet of the phospholipid bilayer. So this is a permeability barrier. Small nonpolar molecules can often cross because they can penetrate the hydrophobic interior. Larger molecules or charged molecules typically can't get across without assistance of a protein-based channel, some sort of transport system. Water can diffuse across, but hydrogen ions, for example, protons cannot easily go across. And larger molecules like sugars or amino acids or certainly proteins can't easily diffuse across this unless there's special transport systems allowing that. So that actually gets me to the part of chapter 2 where it's talking about transport systems in the membrane. How do molecules get across here? The general principles are the same as you've learned with other cellular systems, which is that you need, as we said, a protein channel. It can be highly selective or relatively non-selective. I'm going to stop sharing here just so I can show you on the board here for just a second. So if we have our membrane here, then often we have transport systems. Obviously, this is a very idealized just barrel here where things can come across, right? So if you have a concentration gradient where you have a lot higher concentration of something out here than you do in here, then it may be sufficient to have this concentration gradient drive transport. This is the channel through here. And this transport system, in this case, where you have a concentration gradient, can essentially just allow for facilitated diffusion, right? And in this case, there's no energy required. Okay, because it's just the concentration gradient that's driving this to equilibrate across the two sides of the membrane barrier here. However, if there is not a concentration gradient, or in fact, if the gradient is working against you, so say there's a low concentration here and a higher concentration in here, but the cell still wants the whatever this is to come across, then you need to invoke active transport. Okay, active transport means that an energy, some form of energy input needed to overcome the concentration gradient. Okay, so so then the question is, what can this energy be that can drive the uptake of a molecule that's going against the concentration gradient? The energy input is typically in the form of either ATP or proton motive force or symport antiport systems. So ATP is of course present in the cytoplasm of a healthy cell, and in those cases you might have a coupling mechanism that would change the conformation of this to allow the uptake, and you would have, say, ATP going to ADP as whatever molecule it was came into the cell. Okay. In the case of proton motive force, in an actively metabolizing cell that's carrying out respiration, at least, it is creating a higher concentration of protons outside than inside, and the uptake of a proton might be coupled with the uptake of your molecule, so that you don't use ATP, but you bring protons into the cell. You break down the proton motive force, but you get the systems in, or the chemicals in that you want. So that's a special example of a symport, where you bring something in along with your transported molecule. An antiport is simply where you eject something at the same time as you bring something in. These are all described in your book, these different transport systems. So you don't need to know them in great detail. I just want you to understand the idea that if you, there's no free lunch in the business of getting things across the membrane. Either they have to be at a higher concentration so that they're so that essentially entropy will assist them in moving across the membrane if there is a path for them to move, right? If there's a diffusion channel for them to move, or you have to invest energy in it in the form, usually, as I said, of ATP or proton motive force. Okay, I'm gonna go back to sharing the screen now. Okay, so the next point that I wanted to make is that phospholipids are not the only lipids in the cytoplasmic membrane. Bacteria have sort of a functional equivalent of sterols or cholesterols that are used in many eukaryote membranes, not all, but these have a sort of a stabilizing function in the membrane. So bacteria use these molecules called hopanoids. Again, they're not always there, not there in every species, and different species may have slightly different hopanoids. The point is that bacteria, like our own cells, well, not so much our own cells, but like many eukaryote organisms, often tune the composition of their membranes so that it has the sort of the stability or the fluidity that it wants for whatever environment it happens to find itself in. Okay. Sorry, lost that there for a sec. I want to go back to the last slide here for this section, which is that cytoplasmic membranes aren't just mediate transport processes, control what gets into the cell. They also, of course, are involved in the energy metabolism itself. Respiration uses electron transport to set up that proton motive force, where it can then capture that energy in the form of ATP. Of course, membranes are also involved in the photosynthetic light capture process. The proton motive force is used to drive the flagellar motor, which is embedded in the cytoplasmic membrane as well. So there's a lot of roles in energy metabolism of the cytoplasmic membrane. And then, of course, remember last week we were talking about how a key function of cells is the ability to sense and respond to the environment. So because the cytoplasmic membrane is one of the sort of the interfaces between the bacterial cell and the environment, many sensory systems are embedded in that cytoplasmic membrane. And those sensory systems can connect to changes in gene expression or other adjustments that the cell makes in order to sort of optimize its ability to grow and function in a particular environment. Okay, I'm going to stop this particular part of the lecture now.